Dear colleagues, thank you for the interest to my presentation. Here I will provide you with a short research update on our research on bacterial lipoxygenases. This research update was inspired by the recent discovery of Margarita magnifica, the biggest bacteria in the world. In our previous paper, we have found a phylogenetic and statistical association between lipoxygenase enzymes and multicellularity, both in bacteria and eukaryotes. This phylogenetic tree on the slide illustrates this association. The statistical and phylogenetic picture observed was created mainly by cyanobacteria. They are green. Myxobacteria, they are depicted in red. Dictyostolid slime molds, not depicted here, and complex multicellular eukaryotes depicted here in violet. But there was one more bacterial taxon forming one thin subtle branch, Beggiatoa. They are here in the round. It is a very subtle branch, and the statistical contribution of this taxon is such a subtle too. Beggiatoa is a giant sulfur bacterium. Here you can see them on the slide. The yellow spots are the inclusions of elemental sulfur, but as I said on the previous slide, their statistical contribution was so low that we omitted them that time and excluded them from the statistical analysis. But the discovery of Margarita magnifica, the biggest ever bacterium that is bigger than a fruit fly, urged us to revisit our recent bioinformatic findings and return to Beggiatoa because they are the closest relatives of Margarita. We have considered the question why only Beggiatoa possesses lipoxygenases if they are linked to multicellularity and other Teotrichales do not. And we suggested that Theoplocobacteria rely on their motility. Margarita magnifica relies on its eukaryotic-like complexity, and only Beggiatoa has one strategy to breach the substrate pools, its multicellularity. It could explain why it needs lipoxygenases in contrast to our representatives of this order. We have published a research update discussion article in Biochemistry Moscow and a related blog post on Springer Nature research communities. Please refer to these materials for further information. And if you are watching this presentation in recording, you could find this poster on my ResearchGate. Thank you for your attention, and I would like to ask you questions.